Okay, let's continue. In addition to these formulas that we can derive, we can also use a completely different formula which is inspired by the methods of building Blossom 62 or PAM matrices. So, which is using the log-odds ratio. The log-odds ratio is this. It compares the occurrence of an amino acid at a specific column with its background occurrence frequency. That's the odds that we are comparing, and that's the ratio. So this PA is the background frequency of an amino acid. It occurs, for example, if we had an equal percentage of occurrence in nature, that would be PA would be 1/20 for every amino acid. However, some amino acids like glycine or some amino acids may be more frequent compared to other amino acids, like cysteine is a rare amino acid. So these percentage of occurrences may not be equal. So this formula takes into account that fact. In a certain column, if we see a rare amino acid more than its expected occurrency, this is incorporated into this formula. This directly at every position U, we compute the frequency of occurrence of that amino acid A in that column, in that position U. And we compare it with its background occurrence frequency. And if this ratio is going to be something greater than 1, it's going to be a positive number. Otherwise, it's going to be a negative number. Here, if sufficient data is available, this QA is going to be directly, we can use QA as FUA, okay, the frequency. However, so this means that sufficient data means that if we have lots of sequences in our family, and they are very long, okay, and we have hundreds of sequences in our family, we can use this log-odds ratio safely. However, if we have like only 5 sequences or 10 sequences, something dangerous is going to happen here. If this QUA, if the actual frequency of occurrence of a certain amino acid is 0. In the previous case, in the previous formulas, they just accumulate to their contribution to the formula was just 0, so they don't contribute anything. However, if something has a 0 occurrence, it will cause this to be log 0. What is log 0? Minus infinity. So it's going to minus infinity at a position specific, at a certain position. Minus infinity means that if there's a sequence which contains that specific amino acid, or if there's another sequence which does not agree with your profile, when you are doing your dynamic programming alignment, for that letter you are going to have a penalty of minus infinity, okay, for that match. Which means that minus infinity is going to cause rejection of that sequence from the family directly. So in order to solve this problem, one idea is to use pseudo-counts. Pseudo-counts is this. Just imagine, in addition to your sequences in that family, at each position, you assume each amino acid occurs once, okay? These are, you just insert some virtual, non-existent counts for each position. For each amino acid equally, you introduce all these possible amino acids, A, C, D, E, F, G, at each column. So it's like this. We have 20 new sequences, and these 20 new sequences, they are like this. The first sequence is A, whatever, how many positions you have. The second sequence is C, 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 C. So we have some imaginary protein sequences like this added to our profile just to, and their main reason is that in our real family, if at a column, if a specific amino acid like this A it does not occur in this column, we will have minus infinity score there. Just to prevent this situation, we assume that A occurs at least once in this column. This solution may seem absurd to you first, for example, especially when this n, this capital N, is less than 20. It means that it's like you're putting a lot of noise in your family. Okay, all these non-existent sequences. I mean, if you have a family of 5 sequences, adding these 20 sequences is like a bad thing. I mean, it's adding a lot of noise to this family. However, if this n, if you have hundreds of sequences in your family, if imagine this n is 200, the effect of adding these 20 sequences is going to be minimal. And actually, we are going to also see an extension to this formula, which takes into account the fact that this n, even when this capital N is small, we can, instead of adding 1, for example, we could have added 0.1, 0.1, okay? We could have counted these virtual counts, or they are called pseudo counts. These pseudo counts can be added at a smaller level. I mean, instead of counting them 1, 1, 1, 1, we could have counted them 0.1, 0.1, 0.1, because at the end we are computing a percentage. They don't need to be integers actually. The actual counts are integers here. But the pseudo counts that we are adding does not need to be integers. If your capital N is small, you can have these pseudo counts as 0.001, for example, just to avoid minus infinity. Just add something small there. That's the main goal of pseudo counts. Okay, so this is the original pseudo count by adding each amino acid type by 1 to this formula. These are additional counts, additional virtual counts. And here we can also incorporate the background amino acid composition. And also we can add another constant beta to increase the effect of these pseudo counts. I mean, to decrease their noise effect. Instead of counting them by 1, this beta value shows us tells us you are going to count them as a small number, and this PA here is the frequency. So the sum of PAs is 1. Therefore, the total number of counts that we add to this original counts is the beta. Okay, because the summation of, so it's like this. For all PA, beta PA. This summation, this beta is a constant. If you take this beta out, the summation of all the frequencies is 1. So the total number of counts you have added is beta. And therefore, this is the total number. So compare it with this formula. We add 1. We add 1 for each amino acid type. There are 20 amino acid types, so at the end we have added to our total counts 20 such virtual counts. We have added 20 virtual counts, so our number of sequences increased by 20, m plus 20. However, here we are not, these PAs are not ones, PAs are frequencies. The sum of PAs is 1. Therefore, the things we add, the total of the new pseudo counts we had added is beta. And using PA actually is a good thing. What it does is, we just, in the previous pseudo count formula, even if for rare amino acids, we added 1 for a rare amino acid. And for common amino acids, they're also added 1. There was no difference between rare and common amino acids. So this PA adds this difference between common and rare amino acids when using the pseudo count. So this is the final computation of the frequency of an amino acid at a column, and you divide it with the expected frequency PA to get the MUA. And at the end, we get our position-specific scoring matrix itself is also like a profile. As I said, the main difference is the position-specific scoring matrix from a profile is that in the position-specific scoring matrix, we had pre-computed scores. Okay, all these scores to align for all these different types of amino acids are pre-computed. We could have also stored them as a profile and use and look at Blossom matrix and compute these scores on the fly. These scores could have been computed on the fly when you are doing the alignment. But position-specific score matrix is more efficient if you are going to do this alignment between sequences more than one time. It's just going to be a table lookup instead of computing them on the fly. You just compute the PSSM once, and then you can use it in any sequence alignment. You just do a table lookup. You don't need to go to the Blossom matrix again. It's incorporated within the PSSM. And at the end, there are different ways to visualize such profiles. One visualization idea is, I think it's a nice idea, which is taken from the information theory. It gets some ideas about information theory, is this web logo approach from Berkeley. Here, I mean, look at this. If I just told you, if I give you a PSSM, forget about this alignment at the top, okay? Because after we create the profile, we don't need the multiple alignment anymore, okay? The profile representation is independent of the multiple alignment. It is derived from multiple alignment, but at the end it just shows how much percentage, what is the frequency of amino acids at each column. Viewing this profile with just numbers is not very good, okay? Especially if you think about protein sequences, lots of numbers, it will be very difficult to interpret. At which positions are more conserved, which positions are less conserved. It will be difficult to interpret such numerical profile. So this, to visualize this profile, one idea is to use the entropy idea from information theory. So the entropy is like this. What they do is, for each position, so this is a profile with 20 positions, they compute a weight for the font that they are going to use for that amino acid. For example, here, as you can see, if we had used direct frequencies, if we did something like this, if we directly said that, okay, if you have a profile, if, for example, the first position is 0.5 A and 0.5 C, and the second position is all R, okay, 100% R, and the third position is, for example, L, I, V, M, okay, equals 0.25, 0.25. Now, one way to visualize this is to use different size fonts for each of these letters, so something like this, and based on their frequencies, proportional to their frequencies, I can change a font size to view this. And if you say that my biggest font size is 48 points, okay? So you are going to, since this is 100% R, you are going to have a 48 point R, and these are 0.5, 0.5, so 24 points font C and A, and these are equal 12 point LIVM. Okay, this would be one idea. What is the bad thing about this? The bad thing about this is, okay, the web logo use a similar approach but different. Now I'm trying to tell you the difference of the web logo approach. They do not directly use the frequencies for font sizes. Because if we had used directly the frequencies for font sizes, this profile would appear at same height, okay? I mean, if you add these points at the end, our profile is going to have the total number of points that we are going to have in this representation is 48 points, okay? Because you're dividing this 48 points equally among the amino acids. So they are going to appear flat. Their top height is going to appear at the same level. So what is bad about this, appearing at the same level? So it's going to be a very crowded picture, and the conserved columns will not be immediately distinguishable. So what this web logo does is, if a column is conserved, if there's 100% R in that, use a bigger font for that. I mean, it's, you do not use something flat. If, for example, at this G is very, this column is very conserved, and the idea is conserved columns we want to emphasize those columns by making them larger. So they use an idea from information theory. In information theory, entropy is computed. This HU is the entropy of that column. If you have a varying amino acids in a column, your entropy increases. You have a higher entropy. If you have the same thing, if you have the same signal in a column, if you have all G's in the same column, you have a very low entropy. So based on the negative entropy, they introduced this weight I for each. So they multiply the frequencies. So residual contribution at each position is actually multiplied with this I, which is the information content of that column. So this I does not depend on the residue type, as you can see. It's a measure that you compute for a position. So each position has a different information content. If a position is more conserved, actually its entropy is lower, but we want to take the reverse of that. We want less information. Less varying information is good biologically. So if it has smaller entropy, we take the reverse of it here, and it's going to appear larger. This I is going to appear, IU is going to be larger if that column has a smaller entropy. If more conserved, smaller entropy, and it appears larger in this representation. So this is, again, a heuristic, okay? You could have, I'm sure if I ask everybody, how would you visualize a profile, everybody would come up with different ideas to visualize the profile. And that would be, for example, the colors may also have certain meanings. Maybe polarity, polar, non-polar amino acids, they may use different colors for them. That would be, or based on secondary structures, if we can predict those, we can use different colors here. Okay, now I'm going to start a different representation. So what we have seen here, the profile representation, was just using frequencies at each position. Now, can we have a better model by using hidden Markov models, which were originally proposed for speech recognition? Okay, hidden Markov models were first proposed in speech recognition. And they are probabilistic models. They are like probabilistic finite state machines. And profile hidden Markov model is something specific to bioinformatics. When you see profile at the beginning as a prefix to hidden Markov model, you understand sequence profile. You understand protein families. Okay, so profile HMM is a model proposed for protein sequence family representation. Now, I'm going to start with a general description of hidden Markov models. What there? Yes, sure. Sequences. We do sequences. So we categorize, we have sequences and we organize them. We categorize these sequences into families by first sequence similarity. By putting similar sequences into a family. And then the next step is to have a representation. Yes, exactly. What is the profile of a family? If you have these sequences, if you have these 100 sequences categorized, grouped in this family, how do you represent them? Can you produce a single representation for that family, apart from these 100 sequences? 100 sequences on top of another, they may not be very meaningful to interpret. So can we have a model, can we have a representation for that family? That's the main goal. So one disadvantage of the profile model, the just numerical model, was that even if certain unimportant regions, they take equal space with important regions in representation. Okay, there were unimportant regions which were, for example, look at this. This region is not very important all the different amino acids can be seen at that point. They are not, these two positions are not very well conserved, and there are also some positions which you can see almost any amino acid in that column. These positions are not very important. However, if those positions are not very important, do you really have to put them in the profile? Can you just say, okay, you can have anything there, or you can have two? So this is the main problem in this profile representation. I mean, every position is accounted for at the multiple alignment, but at the end, our model does not emphasize the conserved regions more. So we can have maybe a much more much more compact model, which just takes into account the conserved regions, the conserved blocks, columns, and provide the representation based on these conserved parts. Like, is there a certain pattern that is apparent in this family? Maybe a certain conserved 5 amino acids is the main thing that distinguishes this family from others, and we should bring that pattern, that conserved columns in this picture. So let's start with the general description of hidden Markov models. Now let's go outside biology, outside bioinformatics, and describe what a hidden Markov model is. And at the end, you are going to see they are very simple things. Okay, they're easy to interpret, easy to work with. They are nice models. And here, this shows a hidden Markov model. So a hidden Markov model lambda is determined by the following parameters. It has certain parameters. The first parameter is a set of finite number of states. So we have states in a hidden Markov model, and a finite number of them. And these states indicated here are with circles. So these x1, x2, and x3 are our states. So we have finite number of states. And we also, our hidden Markov model, we are going to simulate a temporal process. We are going to start with something, we are going to go from state to state, we are going to produce some output. So hidden Markov models are Markov processes. A Markov process is a temporal model. So there are discrete time steps, and at these discrete time steps, some things happen. And since we are dealing with discrete time steps, we have a starting time. Okay, so we start this process at time t is equal to 0. And the question in hidden Markov models is, at time t0, in which state are you going to be? So this is your starting state. And given a hidden Markov model, another parameter is the probability distribution of starting at states. So we have 3 states here, x1, x2, x3. Such a parameter would be, for example, x3 0.8, x, or for example 1.0, 0.0, and x1 0.0. So this is a probability distribution. Okay, if these numbers add up to 1, we have a probability distribution. So the probability of starting at state x3 is 1. Sorry, let me make this x2. X2 is 0 and x1 is 0. So it means that at time t1, we are 100% sure that we are going to start at state x3. This could have been something else, any probability distribution. We could have something like this, 0.3, 0.3, or 0.4. It's almost equally likely to start at any state. Okay, we have a starting probability. These are indicated as SI. These probabilities are PIs, the starting probabilities. Our states are SIs in this formal definition. And we have arcs. We have directed edges or arrows. In graph terminology, it's directed edge, or you can tell them arcs, or you can tell them arrows. So there are actually, this picture quality is very bad actually, but there are arrows in these edges. Okay, from this A12 shows the transition probability from state x1 to x2. A21 shows the transition probability from x2 to s1. So the transition probability is this. If at time T1, okay, we are in this state. Where will I be at time T1 plus 1? Okay, if I increase the time step by 1, which state are you going to go from that state in the next state? These transitions can be self-transitions. You can, in the next time step, you can still stay at the same state. So you can, you don't need to go to another state. You can again visit yourself in the next state. But we need these are called the probability of going from one state to another in a time step is called a transition probability. And we need the transition probabilities between every state. So in other words, we have a matrix, the transition probability matrix between each state, and self-transition probabilities are going to be indicated at the diagonals, in the diagonal. And also at each state, we do some output, we do something. When in the beginning for a certain time step, if we arrive at a certain state, we emit a symbol. Okay, the symbols have a finite alphabet. They have an alphabet. Okay, a symbol omega is emitted in a state Si, and as you can see, the emission probability density. This shows that we do not emit deterministic symbols. In other words, the emission is also probabilistic. In a state, we can emit multiple things, but one at a time, okay? We cannot emit multiple things at the same time step, but multiple amino acids or multiple nucleotides can be emitted in a state with different probabilities. A state may emit different things at different times. And this is indicated by the emission probability. What is the probability to emit this, to output a certain symbol in a state is indicated by the emission probability. So if we are given all these things, if all these parameters are specified, what we have ourselves is a hidden Markov model, okay? A defined, known hidden Markov model. Now, our problem is going to be given a multiple sequence alignment similar to the profile representation. We started with this multiple sequence alignment. Given such a multiple sequence alignment, how can we construct a hidden Markov model for this multiple sequence alignment or for this family? We are going to constructing a hidden Markov model means that finding out the values of these parameters. The first thing is, how many states are you going to have in your hidden Markov model? And in which state you are going to start from? What is the transition probabilities from state to state? And the first thing is, if you're going to model, you use the hidden Markov model to represent protein sequences, a family of protein sequences, what are your states going to represent? Okay, semantically, what's a state? What does emission mean? So you need to answer these questions in order to be able to use HMMs to represent the profile. We are going to come to that, and we are going to first answer what is hidden here. As I said in this description, there is nothing hidden, right? I mean, all the parameters I have shown you. So why do we call it a hidden Markov model? We are going to see it in an example. Here, what is hidden is, we usually use hidden Markov models to model a temporal process. Temporal process means that a process that changes with time. And we can observe the output of this process, but we do not know the actual underlying mathematical, physical model that produced this output. I'm going to give you an example. The same example I have given in years is the zebra model. They use actually the hidden Markov models to study zebra behavior in Africa. Okay, so the thing we don't know here is that the hidden thing to us is what the zebra thinks. Okay, we cannot talk with the zebra and say, okay, what do you think right now? How do you feel right now? So this is what is the mood? What is what are the ideas that are passing through the zebra set? We do not know. That is hidden to us. But zebra is doing some action, right? For example, sometimes zebra is running, sometimes drinking, sometimes eating, sometimes hiding. sometimes sleeping. So these are the things that we can observe. As a biologist, for example, you observe these things. These are the things that you can observe. And these things that you can observe are the output signals Y1, Y2, and Y3 here. Okay? These are as a biologist, we can observe Y1, Y2, Y3. And for a specific zebra, for example, we have seen that Y2, Y2, Y3, Y1, Y2, Y3. We may observe a specific sequence of outputs. Okay? And this specific sequence of outputs may be more than the number of states. Okay? For example, the number of time points, the observations, is not limited with this. The hidden Markov model may produce an infinitely many long output sequence. Okay? Because of these arcs, there are loops in these arcs. You start with Y1, you again produce Y1, Y2, Y2, Y3, Y1. So you can have a long sequence. And the question is, okay, you have observed these long sequence, but which states have you visited during these output that is hidden to us? Okay, we are usually, what we do is, as a biologist, we observe a zebra, and this is what we get. For example, we observed a zebra for 1 week. We said that, okay, running, sleeping, eating, drinking, eating, running. Maybe for 1 day, it ran a lot, okay? Running. It may, for each time it may have run. And maybe the other day it slept a lot. Or, I mean, its behavior may change from day to day. But the thing we do not know is that, why did he drink that much during that day? Okay, what was so what we have here, our hidden Markov model, is our states are zebra's brain model. I mean, or mood, zebra's mood, or you may say zebra's feelings. Okay, whether it's afraid, it's hungry, it's sad, happy, all these things. A state is a mood of a zebra may be modeled as a state, and you may transition from state to state. For example, you may be thirsty, but if you drink some water, you may go from thirsty state to maybe a sleepy state or happy state. Okay, so, but we do not know, we do not actually know these states. If you do not plug some things into zebra's brain, we cannot know. Our only thing we can do is observe its output. And the best thing we can do about predicting zebra's behavior is just prediction. I mean, we can just predict the zebra behavior by the output. So this is, the mood is hidden to us. The actions are observable. Okay, so that's why. So there's some logic has to be there in zebra's brain. Okay, zebra. I mean, if it's not a mad zebra. I mean, if it says there's some, it's a normal zebra. There must be some logic behind these actions, and our hidden Markov model has to capture this logic behind these actions. For example, from a thirsty state, if it's a normal zebra goes to a happy state, it's a normal zebra. But you may have a different hidden Markov model. From a thirsty state, you drink some water and you go to an afraid state. I mean, by drinking some water, you shouldn't be afraid of anything unless otherwise there's some other factors. But this logic, that different hidden Markov models may model a different logic, and that's the goal of hidden Markov models. And we usually ask 3 types of questions regarding hidden Markov models. I mean, they are used to solve different problems. Some of the problems actually I mentioned during this example. The most widely used problem is that if I have a sequence of observations, which states produce these observations? This question is one of the most widely used questions. In other words, you observed a zebra for 1 week, and what you ask is, in this 1 week, predict what was the mood of the zebra in this 1 week. For example, happy, angry, hungry, thirsty. So what is the actual mood? Predict this mood that produced these actions. What could be the hidden states that could have produced this output. And the answer to that question may be multiple. So there is no, as you can see, there may be, since each state may emit multiple symbols. For example, a zebra may be running when it's afraid, and it may be running also when it's happy. Okay? The same action, running action, can be observed both in happy and in running mood. But maybe when it's happy, it's less likely to run. But when it's afraid, it's more likely to run. The probabilities may be different, but you may still observe the same effect, same output from different internal moods. So multiple paths may be possible through this hidden Markov model. Multiple different paths may produce the same sequence of symbols. So if multiple paths are possible, what we should ask is that out of all these possible paths that could have produced these sequence of symbols, which path is the most likely path? Okay? In other words, which was the most likely, most probable sequence of moods that could have produced this behavior, given that model. So this question can be answered given a model, given a hidden Markov model for that zebra behavior, given that logic, and given a sequence of observations, find the sequence of states that produce these observations. So this can be found so this is the second question. Okay, find the most likely sequence of hidden states that could have generated a given output. We can use the algorithm, a dynamic programming algorithm called Viterbi algorithm, to solve this problem. We can also ask this question. We may not wonder whether which internal model produced this sequence. The first question we can ask is that we observe something, for example, is this a normal zebra? This sequence of actions, for example, is this how likely is to observe these sequence of actions? It can be for example, to give you an example like this, we may have two zebra colonies living in different parts of Africa, and they may have different hidden Markov models, okay? Because there may be different threat levels in different areas in Africa. Some of them, their transition probabilities, their emission probabilities, their actions, they may have different behaviors. Okay, in different regions in Africa, these two zebra colonies, they may have different behaviors. Now, we may have a runaway zebra, okay, which we found just in the middle of these colonies. And by observing the behavior, we may try to ask, which of these colonies does this zebra belong to? Okay, so what we would do is we observe this zebra, this runaway zebra, 1 week, and we just ask, okay, and we have two hidden Markov models. Okay, this zebra, these actions, how likely these actions have come out of this hidden Markov model? Okay, and how likely it has come out from the other hidden Markov model? By comparing these two likelihoods, we may say that okay, this zebra probably belongs to this colony or this colony. Okay, this is the first problem. Given the parameters, given the hidden Markov model, compute the probability of a particular output sequence, particular observation. What is the probability of observing this output from the hidden Markov model? And this is also it's solved by the forward-backward procedure, which is exactly the same as the Viterbi algorithm. There is very small difference between these two algorithms. They are both dynamic programming algorithms. And the third question, so we are going to, in this class, we are going to see how we answer these two questions. The third question is a more difficult question. It's this: by observing all the members of the colony, okay, for a week, produce the hidden Markov model for this colony. You do not know the hidden Markov model. How do you get the hidden Markov model for this zebra colonies in the first place? The third question tries to answer this question. If you have a set of observations and you assume what this third question does not answer is that it does not answer how many states you are going to have, which state is going to represent which, what semantically. So this is this model design of the hidden Markov model, how many states you are going to have, what are those states going to represent. That design is completely left to the developer. It's not done automatically. It's not answered by this third problem. But if you know the states, by observing lots of zebras for a period of time, finding out the transition probabilities, finding out the emission probabilities that could have generated these lots of zebra behavior, most likely is done by the Baum-Welch algorithm. It's an expectation maximization algorithm which tries to iteratively finds the best transition probabilities, best emission probabilities that is going to describe your data set best. Okay, this is also called the training phase of the HMM. So you observe, given the given the set of sequences, you can use this to find the hidden Markov model parameters. I'm going to talk about, even if we are not going to talk about the Baum-Welch algorithm, I'm going to talk about the technique. Given a multiple sequence alignment, if we are given a multiple sequence alignment of sequences, we may not need Baum-Welch algorithm to construct HMM. Okay? Because the multiple sequence alignment is a step which answers a lot of different things, like how many states you are going to have, your state transition probabilities. All these can be answered by inspecting a multiple sequence alignment. So, but this Baum-Welch is used in general, not only for protein sequences. It can be used for the zebra problem, for other problems. If you have a set of observations, you can use this to generate your hidden Markov model. Okay? So I'm going to stop here, and we are going to continue with a general example, and then we are going to see general structure of profile hidden Markov models next week.